Today, I will be discussing the results of the synapsis of two different individuals' chromosomes, with crossovers occurring, as well as tying in an example of a chromosomal aberration and what the gametes will look like. I will be illustrating this concept with hand-drawn and labeled images, where the letters represent a specific region of the chromosome and the black circle or dot will represent the centromere of the chromosome. Starting with the first individual's chromosomes, we will want to have both of the ends of the chromosomes matching. To do so, we will need to flip or rotate the bottom chromosome 180 degrees. We can now see that the middle section of the chromosome contains a chromosomal aberration, specifically an inversion. Inversions have a segment of a chromosome that is removed from the chromosome, turned 180 degrees, and then reinserted back into the chromosome. Because the flipped region contains the centromere, this is considered a pericentric inversion. This is an example that explains synapsis during meiosis and the formation of an inversion loop. The original chromosome is represented by the black lines, whereas the lines in brown represent the chromosome with the inversion. For both individuals, we will have a crossover between J and K. We will look at what chromatids are created from this event and discussing whether or not it would produce a viable gamete. This chromatid formed will produce a viable gamete. The chromatid formed here will also produce a viable gamete. Both of these gametes are not missing any genetic material, nor do they carry extra copies of genetic material. With this chromatid, we can see that it has several copies of the gene M and L, making its gamete non-viable. The same instance occurs for this chromatid, which would have two copies of H and G, making the gamete non-viable. These two recombinant gametes will be non-viable with missing an extra genetic material. 50% of the gametes will be non-viable. These two chromatids would produce viable gametes. In conclusion, individual 1 would have 50% non-viable gametes and 50% viable gametes. Going back to individual number 2's chromosomes, we will once again flip the bottom chromosome so that both of the chromosomes ends match. After flipping, we can now see that the two chromosomes are identical and do not have any chromosomal aberrations. Let's go ahead and look at the crossover in individual 2's chromosomes during meiosis. The red X represents the location of the crossover. Because the two chromosomes are identical, the synapsis of the chromosomes will pair normally. This results in 4 identical gametes that do not contain any chromosomal aberrations. All gametes will be GHKJ centromere ILM. Individual 2 would produce all viable gametes, whereas individual 1 has 50% non-viable gametes. This makes individual 2 have a higher fitness than individual 1. Thanks for watching.